All right, welcome. So today we're talking about comparative genomics and specifically evolutionary signatures for genome annotation. So we are entering the last module of the class on comparative genomics and evolution. Today's lecture is going to be on comparative genomics and evolutionary signatures. Next time we're going to be talking about genome scale evolution and genome duplication. Then we're going to talk about phylogenetics and phylogenomics. And then we're going to have the quiz, and then we're going to transition to the future directions. So we've talked a lot about genome annotation, about gene expression and epigenomics, about networks and regulatory genomics, and about disease genetics and disease genomics, which was all about variation within the human lineage. Today, we're going to be talking about variation across species. And specifically, we're going to be looking at how we can use genome-wide studies to infer signatures of evolution and then use these signatures to annotate elements. And we're going to apply these signatures to annotate protein coding, known coding, and microRNAs and regulatory motifs. So protein coding genes, known coding genes, microRNA genes, and also regulatory motifs. And next time, we're going to be talking about genome rearrangements and genome duplication, about how to construct global alignments based on synteny, and how to detect structural changes. And then we're going to have 2 lectures on phylogenetics and phylogenomics, namely, how do we use phylogeny to understand evolutionary rates, models of evolution, distance-based trees, and also we're going to go back to dynamic programming on a tree and Bayesian models of evolution. And then with phylogenomics, it's going to be very similar to today's lecture on using global signatures to understand individual local events. And we're going to be looking at gene trees and species trees and reconciliation and coalescence. So I showed you this slide earlier on to motivate genome alignments, to how to construct alignments. And this lecture basically picks up very much where that left off, namely the idea that we can use alignments of closely related species, which we can construct using dynamic programming, using BLAST, using all kinds of tools for building these alignments. And once we have constructed these alignments, we can go in and look at the conservation patterns of different regions. And here the stars denote perfect conservation across these 4 species that roughly span the distance of human, mouse, rat, and dog. And you can see that there are islands of conservation which stand out based on stretches of stars. And those islands of conservation, in fact, correspond very well to known regulatory motif instances within these regions. So the idea is, can we now use comparative genomics to study these signatures systematically and to discover these elements? And in particular, today's lecture is going to be focused on reading evolution to reveal functional elements. And then next time, we're going to be looking at how we can use genomes to understand evolution itself. So basically, we can, the better we understand evolution, the better we understand genomes, and the better we understand what's in the genomes, the better we can model evolution. So the goal for today is to first look at nucleotide conservation and evolutionary constraint, then to look at evolutionary signatures, which focuses on the patterns of change rather than the amount of conservation. And then we're going to look at signatures of protein coding genes, and then signatures of RNAs, motifs, transcription factor binding sites, and microRNAs, and also how to measure selection within the human lineage. So comparative genomics is extremely powerful. We can use it to compare related species in order to discover functional elements. And the concept is that ever since Darwin, we have understood that the evolutionary process consists of 2 components. On one hand, random mutation. On the other hand, natural selection. And the random mutation is basically exploring space by randomly mutating elements, which is not, trying to do so. It's just making mistakes as it's copying DNA. I like to often joke that if engineers were in charge of selection, we would still be perfectly replicating bacteria with no mistakes. So mutation is a double-edged sword. On one hand, it introduces problems, but on the other hand, it introduces novelty. And what we're going to be focusing on mostly today is how how purifying selection works, namely those mutations that disrupt elements, how those are maintained. On the phylogenetics lectures, we're going to talk a little bit more about positive selection, but now we're mostly going to be focusing on random mutation that mostly breaks things, but every now and then it makes things better. And then natural selection is the brutal decision, which is selecting within the context of the environment those resulting sequences that are maintaining the function. So again, on one hand, we have random mutation, which is completely blind, shoots at everything randomly, and then natural selection, which is maintaining those sequences that maintain the function, that still preserve the function. What is the result of those two forces applied over evolutionary time? On one hand, non-functional regions will accumulate mutations and will be kept. Basically, will accumulate mutations and those mutations will be kept. So over evolutionary time, any mutation that happens in a non-functional region is preserved. But within functional regions, mutations accumulate just as much, but they decrease fitness. And therefore, over evolutionary time, it will lead to those organisms that have disrupted elements to basically eventually become less fit, and their genes will eventually thin out. So if you look at the effect of all this is that even though the same amount of mutation happens in both types of regions, in both functional and non-functional regions, over evolutionary time, those mutations in functional regions are excluded from the gene pool, and those mutations in non-functional regions are accumulating. So who's with me so far on this concept of random mutation followed by purifying selection? Awesome. So 67-33-000. So one of the things that we care about is, of course, power. So how much power do we have to discover these functional elements? And the very simple concept is that the more branch length you have, the more total length you have on the string, the more events you have that will basically be accumulating in non-functional versus in functional regions, and the more power you will have to distinguish the accumulation of mutations in the non-functional regions and their exclusion from functional regions. So your power should be scaling roughly linearly with the amount of evolutionary distance that you capture in the tree of species that you're aligning. And if our goal is to distinguish functional versus non-functional based on the number of mutations, with very close distances, there's really no mutations in either region. With sufficient distances, we can distinguish the differential accumulation of substitutions in functional versus non-functional regions. And with very, very distant comparisons, then the functional regions are no longer conserved. So the challenge there is that, if you compare, amoeba and mammals, there's very little conservation, and therefore you won't be able to really capture this distinction between functional and non-functional. So the solution that we often take in comparative genomics is to compare many closely related species. And that's much more powerful than few distantly related species. Namely, for the same total branch length, we prefer many close species because the functional regions are conserved for each pair of species. They haven't had enough time for any pairwise evolutionary comparison to accumulate enough new environmental adaptation. Basically, the environment hasn't changed that much in short evolutionary distances. And therefore, the pairwise conservation will be still high, but the non-functional regions will accumulate noise independently. And the analogy that I like to give here is when you're recording an orchestra with one microphone, If there's noise surrounding that microphone, you're going to record as much noise as signal. If there's multiple microphones, the signal will be transmitted linearly to all of the microphones, but the noise will be independent in each of the microphones. And therefore, we're able to actually capture the evolutionary signal because of this independent accumulation of noise in the different lineages. So everybody with me here on this concept that we want a lot of branch length, but the way to get this branch length is much more powerful when you look at multiple closely related species rather than at just two very distantly related species. Great. So 67, 17, 17, 00. So now we have studied the concept of neutral branch length and how it scales with discovery power and how purifying selection maintains functional elements unchanged by excluding those species and those organisms that have accumulated mutations that disrupt them. Now let's look about detecting constraint elements and specifically looking at individual nucleotides or windows or building a hidden Markov model that can detect these nucleotides. So you can build basically now these genome-wide alignments that reveal orthologous segments. And for any one species, you can see the genes on the Watson strand or the Crick strand in different colors here. So some are encoded facing to the right, some are facing to the left. And then over evolutionary time, you can basically align these species and see that the order is roughly conserved. Why is that important? Because by choosing species where the chromosomal segments are roughly conserved in the same order, that means that we can construct alignments not just of the protein coding regions, but also of the intergenic regions in between these genes. So that means that we can actually study patterns of evolution throughout the genome in both coding and non-coding regions. And this is true across hundreds of genes at a time. So we can now start using these comparisons to identify functional elements within them. And I showed you this slide also in the very first lecture, or lecture number 2, looking at comparative genomics and how it can be used to reveal functional elements by showing that, for example, exons, these are protein coding exons in tall blue lines, are in fact very deeply conserved. And the challenge, of course, is that many other elements are also strongly conserved. And the question is, are these in fact exons or regulatory regions? So if we're trying to understand whether this particular region here is coding or non-coding, we can actually, I don't know what's wrong with the animation here. There you go. We can actually ask, well, what is this region? Could it be another protein coding gene, exon, that is just simply not annotated? Could it be a long non-coding RNA? So if we look at the patterns of change of this sequence, we can basically see that, the overall length of the phylogenetic tree over which it is conserved stops outside mammals. So it's not conserved in chicken, fugu, or zebrafish. So that suggests that this might actually be a non-coding region because protein coding exons tend to be very deeply conserved even outside the mammalian tree. So in general, protein coding genes evolve, much slower. And we can now ask, well, great, let's first of all develop methods for detecting constraint. And then we're going to develop methods for distinguishing different classes of constraint. So for detecting constraints, we talked about how we can count the total number of edit operations or the number of substitutions and gaps. And that's what our dynamic programming lecture was about. Or we can estimate the number of mutations, including an estimate of back mutations. And that's going to be the focus of our phylogenetic lectures, lectures 3 and 4 of this module. We can also incorporate information about the neighborhood. We can look for conservation windows. So as we go, for example, in different stretches of nucleotides, we might say, okay, well, here that's a conserved nucleotide, but it's conserved alone, and that's another conserved nucleotide, but it's conserved alone. Maybe the fact that multiple nucleotides in a row are conserved is actually important, so we can use windows of conservation to detect those. So that's sort of the concept of windows, and you can write a hidden Markov model, for example, that searches for the best transitions between highly conserved versus lowly conserved regions. And we talked about an application for hidden Markov models in the HMM lecture for understanding where are islands of protein coding constraint or where are islands of non-coding constraint in the human genome. And in particular, the information that you see here on the y-axis shows the posterior probability of being in a hidden Markov model state that is corresponding to conserved versus non-conserved elements. So again, you can estimate the probability, the posterior probability of a constrained hidden state, and you can use either Viterbi decoding or posterior decoding. You can use Viterbi decoding to find the path that best corresponds to the best parts of conserved, non-conserved, or you can use posterior decoding to define what are the most likely hidden states for every single nucleotide in that region. And again, you can use phylogeny to estimate the tree mutation rate or the rejected substitutions. And that's basically asking, along my tree, are there individual SNPs that have individual mutations that have not occurred when I would have expected them to occur? And we could also allow different portions of the tree to have different rates. And we're going to talk about that in the phylogenetics lecture. But the main thing that I'm going to be focusing on today is the concept of patterns of selection versus rates of selection. Here are 3 examples of regions that are evolving neutrally or non-neutrally. So in the neutral region, you see that there's a lot of substitution. So every conserved position is shown as a dot, and every change is shown by the nucleotide to which that sequence has changed. So C, for example, has changed repeatedly in T in this entire subtree, and then into A for this lineage and to G for that lineage, and into A for that entire subtree, and so on and so forth. Okay, so a neutral sequence basically has multiple independent changes that are switching between every nucleotide and every other nucleotide. And that's basically, the neutral sequence. The constraint sequence can basically show a decreased rate of change. That's basically saying, well, great, I can have any kind of mutation, but I just have fewer mutations overall. So there's many more dots, there's many more constrained sequences. So we can build a probabilistic model of a substitution rate that has a maximum likelihood estimation of that rate of evolution, omega, and we can report the rate omega, and also report the log odds score that rate is non-neutral. And then we can use either a window-based or a site-wise application of omega. Everybody with me so far? Okay, I see a question. How does branching occur? So the branching is basically speciation. So that's when, for example, this species here was separated from that species here, either because of a geographic event or because of a, post-zygotic segregation event where, for example, there may have been a rearrangement in one of the chromosomes of the ancestor of Ananasi that basically makes their chromosomes no longer line up. So even though the flies might be attracted to each other and, have offspring, that offspring might not be viable because they can't line up their chromosomes properly. Another possibility for this branching is, for example, in the Hawaiian flies, my uncle, Michael Cambiselis, who was actually a professor over at NYU, he actually studied the speciation of the Hawaiian Drosophila species. And what he found is that, for example, flows of lava could actually separate species, and they would no longer be able to mate with each other and then lead to speciation events. So that's how these branches occur. They can either be geographic isolation or post-zygotic, which basically means they can have zygotes, but the zygotes are not compatible. I hope that answers your question. All right, so we talked about the intensity of constraint, but then we can also study this region here. And what we find in this region is that there are a lot of independent events where the C change into a G, and again into a G, and again into a G, and again into a G, in a bunch of different independent lineages. So these are not phylogenetically congruent as you have here. Instead, they represent independent events. So what does that tell us? Does that tell us that this nucleotide is evolving randomly? Is it neutral evolution? Well, no, it's not quite neutral. It is constrained to be either a C or a G. Half the species have a C, half the species have a G. So instead of basically studying a rate, which here would be very high, suggesting that that nucleotide is evolving neutrally, we can instead detect unusual patterns of substitution. We can basically build a probabilistic model of the stationary distribution for a Markov chain between the 4 characters A, C, G, T. And then as the letters are switching in that Markov chain, we can basically ask, are they switching to each other in a non-random way? We can basically build a maximum likelihood estimator of this vector of what are the nucleotides that are being constrained at that region, and then report a position weight matrix for every k-mer in the genome and the log odds that that k-mer is non-neutral. Who's with me so far on this distinction between the overall amount of change and specific patterns of change? Awesome. So 36, 64, 0, 0, 0. So using this, we can now go through the genome and not just say what is highly conserved or what is lowly conserved, but also what is conserved as a T or G, as an A or C, or a T or G, and so on and so forth. Okay, so we can basically match position weight matrices across the genome. And we've used that to now reveal individual transcription factor binding sites and also within motif instances of position-specific biases. And with more species, you can actually derive the motif consensus directly from that sequence, where basically you can see that multiple nucleotides are actually being constrained in a particular location. And you can use that to now recognize genetic variants that are disrupting motifs that are evolutionarily conserved. And we talked about that when we talked about GWAS functional interpretation. So that's about detecting constrained elements, either through the sequence specific, the position specific amount of mutation, or the position specific pattern of change. And now we can also use that to estimate the fraction of the genome that's constrained. Namely, even if we can't detect an individual nucleotide, whether we can detect the fact that across the entire genome, there appears to be more constraint than what you would expect by chance. So the idea here is that we're going to be looking at the distribution of evolutionary constraint. So we can measure constraint either through omega or through pi, through the rate or through the pattern of conservation. And then based on the distinction between these two curves, recognize how much conservation is actually detectable. So that's about putting a threshold here and then asking what lies above that threshold. And at every threshold, we have some false positive rate. So we can choose this false positive rate based on neutral elements. We can basically ask what is the amount of evolution that I would expect for neutrally evolving regions. That's the blue here. So we can basically look at the distribution of conservation scores, that's the red, versus expected distribution if there's no constraint, that's the background in blue. And then at any cutoff, we basically have a set of true positives, which are all the ones that are in the red distribution above the threshold, or false predictions, false positives, which are the ones that are in the blue distribution above that threshold. And at every threshold, we basically have some false positive rate. So the false positive rate is basically telling you, I can choose the threshold based on that blue distribution to have a false positive rate of 5% or something like that. And based on the difference between those two distributions, I can choose a threshold and a false positive rate at any distinction. But the problem is that we can't detect all of the constraint elements because the curves overlap. Basically, yes, the true signal is slightly shifted to the right compared to the neutral signal in blue, but the distributions are still highly overlapping. So, we will never be able to detect something here without also letting in a bunch of false positives there. However, what we can do is actually estimate the total amount of excess constraint by integrating over the entire area between the two curves. And using this, we can basically say what fraction of the genome is not only detectable as constraint, but also what fraction of the genome is estimated as being under constraint overall, even though I will simply have no power to detect those. And what this analysis has led to in the context of the mammalian alignment is that there's a very strong enrichment for constraint within protein coding transcripts, but also in the 5' UTRs and 3' UTRs and in promoters and, in other regions of the genome. But then the new elements are largely falling in intronic and intergenic regions. Namely, there's, yes, less enrichment, but an enormous amount of constraint there. And using this, we can basically estimate by comparing the blue and the red curve that there's this excess constraint in this, purifying selection side. But if you scale up this curve of basically the difference between the whole genome in blue and the ancestral elements in red, and these are ancestral repeat elements, which we expect are not very heavily constrained. So we can use these red elements to basically estimate the excess constraint. And then that excess constraint has in fact two modes. One mode is in fact purifying selection, that's excess purifying constraint. And the other one is actually excess positive selection, where basically there's new elements being discovered there, being evolved there. So who's with me so far on this whole concept of not just being able to detect elements using a particular threshold, but also look at the excess elements, excess constraint across these elements? Okay, so nearly everyone is pretty high except for one person a little lower. If you have any questions, please put them in the chat and I'm happy to answer them. The constraint that we've talked about here has been based on the actual evaluation of constraint across different elements, but you can also look at the overall depth of coverage at different regions. And what you can see is that, for example, protein coding exons have a coverage of about 20 species out of the 29 species that we had been comparing. For ancestral repeats, you have much less coverage. What does that mean? That means that not only are the aligned species highly conserved there, but moreover, there are more aligned species in the regions of the genome that are functional. So you can use the overall coverage depth as an additional measure of constraint. And if you look at elements that were conserved across 4 mammals, you can see that they're much more aligned across the 29 mammals. And then these new elements that we're discovering are also very highly aligned and also highly constrained. And right now we're only using the constrained signal, but there's additional residual signal left in the presence or absence of an alignment at that particular location. So you can see here how the power increases from 4 species, human, mouse, rat, dog, to 29 species. You basically see that we can detect a lot more constraint, but we can also estimate much more constraint. And the constraint here at 50 nucleotide resolution is roughly 5% estimated. But with the patterns of change, we're finding more constraint estimated. Okay. So that's all about measuring constraint at the nucleotide level. But now when we look at larger elements, we can actually look for patterns that involve multiple nucleotides at a time. So now we're going to talk about the concept of evolutionary signatures and then specifically focusing on the patterns of change. So the idea here is that we can basically develop signatures for detecting that perhaps this region here is actually evolving differently from those other regions there. And then the concept here is that the specific function of a region, whether it's a protein coding gene or a non-coding RNA or a regulatory motif, the specific function dictates the selective pressures that are acting on a region. And those selective pressures, in fact, dictate the patterns of mutations, insertions, and deletions that we're going to be finding. So we can therefore develop evolutionary signatures that are characteristic of every type of function. For example, if you look at protein coding genes and you compare protein coding versus non-coding regions, the codon substitution frequencies themselves are telling you that triplets of nucleotides are exchanged at a rate that is consistent with selection for amino acid function, whereas in non-coding regions, triplets are exchanged, roughly randomly. So the first signature of protein coding evolution is codon substitution frequencies, and the second signature is reading frame conservation, namely a pressure to preserve the reading frame of translation, so that the gaps are always multiples of 3, here 6 and 3, as opposed to here 14, 7, and 17. So these are evolutionary signatures associated with protein coding function. We can also talk about evolutionary signatures associated with RNA structure. So here, the function is not at the amino acid level, the function is at the base pairing level. And therefore, those substitutions that change the nucleotide sequence but preserve the pairing will be tolerated. So if you have a substitution that changes a GC base pair into an AT base pair, that's perfectly fine. The folding still happens. So compensatory changes that preserve the folding are in fact a signature of RNA structures. And also silent GU substitutions, where basically G can base pair with either C or with U in an RNA structure. And these GU-GC substitutions are another one of those evolutionary signatures. Notice we're not asking how much conservation is there. We're asking whether the changes are specifically preserving a signature of selection for a particular function. So over here for protein coding regions, the white nucleotides are the ones where there was no change. We're not even using the white nucleotides. We're only using the colored nucleotides, which are the ones where a change has occurred, to basically look for these evolutionary signatures. For microRNAs, the conservation profile is very characteristic of sort of low conservation here and in the middle, but high conservation in the branches. And then the structural features of loops and pairs and a relationship with 3' UTRs that give us additional signatures. And for regulatory motifs, the signatures are that the mutations actually preserve the consensus and they allow for increased branch length score at any one location, and also a genome-wide conservation. So these signatures together allow us to detect regulatory motifs from their evolutionary signatures and also detect individual motif occurrences. So the reason why I'm showing this slide, even though we're going to dive into details for each of those, is I want you to recognize that there's very different classes of evolutionary signatures for every type of functional element. And the key idea behind there is that mutation will randomly explore this evolutionary space, and the shape of that space will be very different for protein coding genes. The shape might look, I don't know, more this way. And for RNA, the shape might look that way. And for microRNA, the shape might look like that. Basically, we're looking at the space of all sequences that all encode the same function. And that space is what allows us to detect these evolutionary signatures. And in that space, you basically have to think about the impact of more branch length. If you are trying to infer that the shape, I don't know, is a square versus a circle, with very few dots thrown at the paper, you might not be able to tell. But with more events, the shape of mutations that are tolerable is in fact much more clear. All right, so that's the concept of evolutionary signatures, the fact that different classes of elements evolve in different patterns, in different ways. And we're not going to be focusing on the amount of change or the amount of conservation. We're going to be focusing on the specific patterns of change. So who's with me so far on the concept of evolutionary signatures and how they're distinct from simply the overall amount of conservation? Awesome. So 40, 40, 25, 0, 0. Okay, so using this, we can now go into protein coding genes and discover new protein coding genes, or revise existing protein coding genes, or detect unusual gene structures. Within RNAs, we can develop methods to detect novel structural families, or ways of recognizing targeting or editing or stability signals, and also ribose switches. We can, within microRNA evolutionary signatures, we can discover novel microRNA families, expand existing microRNA families, look for specific classes of microRNAs where the two arms are cooperating, or where the sense and the antisense strand are in fact both functional. And we can also, in the space of motifs, discover new regulatory motifs, new regulatory motif instances, or networks of transcription factors in microRNAs, as we discussed in the regulatory motifs and the regulatory genomics lecture. And also we can infer single binding site resolution for many of those. So let's dive into signatures for protein coding evolution. So we're going to discuss reading frame conservation and also codon substitution frequency. Okay. So here's the test for you guys. Here's an alignment of 12 species of Drosophila, and these again are spanning a distance similar to the mammalian distances of 29 mammals, about 60 million years worth of divergence. And we can now ask, where is the protein coding region here? So part of this region encodes a protein, and part of this region is non-coding. Who wants to take a guess as to what region is protein coding versus non-coding? You guys can annotate directly, or you can raise your hand, or you can unmute. So if we draw a vertical line here, I will claim that the sequence to the left of this line is evolving differently from the sequence to the right of this line. So what are some of the evolutionary signatures that we talked about that are visible here? The first signature is in the gaps. If you look at the gaps here, these are length 5, and that's length 1. That's not the kind of gaps you would expect in a protein coding region. By contrast, this is a gap of length 6, 6, and 3. And the reason for that is that within protein coding regions, you want to preserve the reading frame of translation. Okay, whereas here, there's no constraint for preserving the reading frame. The second signature is that if you look at the patterns of mutation here, they're actually happening periodically. So conserved, conserved, not conserved, 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 not conserved. What I like to call bang, bang, poof, bang, bang, poof, bang, bang, poof, bang, bang, poof, bang, bang, poof. So the stars are aligned, are arranged in a periodic pattern, and that is largely driven from the silent codon substitutions that are largely involving the third codon position. And more specifically than that, there are specific synonymous substitutions that are happening much more frequently within protein coding regions, but not within non-coding regions. And lastly, you can see that there's a splice site here, GTAG. This is the acceptor site, where we're switching from a non-coding to a coding regime. So these are basically evolutionary signatures for protein coding selection. Once again, notice that we haven't even colored the conserved nucleotides. We're only coloring the changes. So who's with me so far on the evolutionary signatures for protein coding conservation? Awesome. So 75, 13, 13, 0, 0, 0. Okay, so as I mentioned earlier, the frame shifting signature comes from the fact that if you delete an entire codon, for example, the fat cat sat, if you delete that entire codon, the cat sat is still interpretable. But if you delete either one nucleotide, like the F here, that shifts the reading frame of translation of all the subsequent nucleotides and all the subsequent codons. So that basically makes it nonsensical. So the, at doesn't mean anything because you've shifted by one. If you delete two nucleotides, F and A, the, ka, sa, t doesn't again mean anything. But if you delete three nucleotides, you're back in frame. So the insertions and deletions that mangle the remainder of the protein are causing a frame shift, and therefore they're very strongly excluded within protein coding regions. Similarly, for this bang, bang, poof pattern, for this conserve, conserve, not conserve pattern, you can see that the first two nucleotides are sufficient to specify the translation of any of these four codons into the same amino acid. And same thing here. If I have the first two nucleotides conserved, the third one is basically free to vary for the vast majority, or for a big portion of the genetic code. So basically, this table of translation of the genetic code is in fact what dictates this periodicity of mutational constraint. So natural selection tolerates mutations that have little or no effect on the protein. So we can use this to now look at the alignment of a protein coding region versus a conserved non-coding region, and we can color synonymous, conservative, and non-conservative codon substitutions. Synonymous codon substitutions are the ones where the amino acid translation is exactly preserved. The exact same amino acid is translated. Whereas conservative simply means that you're replacing that amino acid with another amino acid with similar properties. And then non-conservative means that you're replacing that amino acid with another amino acid with very different properties. So if we color these synonymous versus conservative substitutions in this alignment of 12 flies, what you can see is that all of the mutations here are in fact synonymous or conservative, and the vast majority of mutations here are in fact not conservative, because in non-coding regions, evolution doesn't care to preserve these synonymous or conservative. They don't mean anything. And if you change a triplet of nucleotides randomly, you're just as likely to hit any of the first or second or third position, and therefore most of them will actually disrupt the amino acid translation. We can also color frame shifted nucleotides based on insertions and deletions that are not multiples of 3. And here you don't see the gaps because we've aligned them to the reference of the ancestor, but these gaps are still colored. The locations that are in the wrong reading frame are still colored in orange. And then lastly, you also see that there's stop codons that get inserted in the middle of this region, because again, there's no pressure not to include stop codon. So even though this region here has roughly the same evolutionary rate as this region here, their patterns are dramatically different. And you can zoom out and do this across the entire genome. And what you can see here is an entire region where you see non-coding, non-coding, and then protein coding. And again, you don't need to be a statistician to sort of see the humongous strength of signal here, separated exactly by this stop codon there. Okay. So these are the signatures for protein coding genes. We talked about reading frame conservation and also codon substitution frequency. So now let's see how we can score these signatures. For reading frame conservation, we can basically ask what fraction of the sequence is in the same reading frame. And over here, you can see that if I break this region up into little windows and I ask what fraction of that window is in the same reading frame, I get 100% in this case, but I get only 60% in that case, given the large amount of frame shifts. And we can calculate that by simply aligning all 3 frames and then asking which frame gets the maximum score by simply saying, well, if I interpreted this as all being frame 1 or all being frame 2 or all being frame 3, what is the maximum of the 3 I can get? And then that allows you to sort of infer this dramatic distinction between those. So using just reading frame conservation within the yeast genome, we were able to see that named genes were in fact accepted at 99.9% sensitivity, and random intergenic regions were rejected at 99% specificity. And these additional 2,000 hypothetical ORFs were in fact classified as either protein coding for most of them, but there were 500 that were very strongly rejected. And here's an example of a very strongly rejected ORF. You basically see here this ATG, the stop codon, and then these patterns of substitutions that are not periodic, and also this large number of insertions and deletions. For the codon substitution frequency, we're now going to develop a likelihood ratio framework by estimating a coding substitution matrix and a non-coding substitution matrix, and then scoring this systematically. So how do we distinguish protein coding versus non-coding constraint? What we're looking for is a method that can quantify the distinctiveness of all 64 by 64 codon substitutions. Why? Because there are 64 possible codons, and any of them can be changed into any one of the 64 possible codons. So for synonymous substitutions, we would expect them to be very frequent in protein coding regions. And for nonsense, for example, we would expect them to be much more frequent in non-coding than coding. And we can model the phylogenetic relationship among the species with multiple apparent substitutions that can be explained by a single evolutionary event with species that are phylogenetically consistent. We want to tolerate uncertainty in the input, where we don't know the ancestral sequences, so we're going to be integrating over all values of the ancestral sequences. And we also want to tolerate alignment gaps, where basically for low coverage species, we might be just missing that part of the genome and also missing data. And lastly, we want to report the certainty and uncertainty of the result in form of a p-value. We want to quantify the confidence that given the alignment, that a given alignment is protein coding. And the units are going to be either p-value or bits or decibands and so on. So how are we going to do that? We're basically going to model evolution as happening along a phylogeny and ancestral sites changing into progressively, different sequences, and ultimately giving rise to the observations we have. We have, of course, only observations for the present day species. We don't have observations for these ancestral species. So we're going to have to be integrating over their possible values. So at every site, which is every alignment column of every triplet, every codon, is going to be treated independently. And then given the tree topology and these conditional probability distribution of evolving under a protein coding or a non-coding model, we can simulate evolution of an ancestral sequence into the subsequent event. And we can, given the leaf sequences, infer the ancestral sequences either through maximum likelihood or through probability distribution over all ancestral sequences. And for L leaves, these conditional probability distributions have a large number of parameters. So it's basically 2L minus 2 times 64 square parameters. But the problem here is that instead of having a different substitution matrix at every branch, we're going to couple them and basically think about a single substitution matrix for the entire tree, therefore limiting that number of parameters. So what we're going to be doing now is parameterizing this conditional probability distribution by a rate matrix, which is actually shared throughout the tree, and a branch-specific time, which is going to be the branch length by which this is scaled. So we're going to have the probability that the child has value B, given that the parent has value A, and that T time has elapsed, as basically a matrix that is raised to the power T. So basically, this is just a rate matrix for an infinitesimally small time T, which you can then scale to the tree of the whole branch. So the branch lengths specify how much time has passed between any two nodes, and the rate matrix describes the relative frequencies of codon substitutions per unit branch length. And the synonymous substitutions have very high rates, and the nonsense substitutions have very low rates. So we can obtain maximum likelihood estimates of now, instead of 2L minus 2 times 64 square, a single 64 square matrix shared across the tree, plus, not times, plus 2L minus 2 parameters, which are the scaling factors for each branch length. And we can use expectation maximization to infer these parameters. So here's an example of a 4x4 substitution matrix and rate of substitution. So basically here we have this 4x4 matrix that basically tells us with what probability is, or what is the score with which each of these nucleotides is replaced by another nucleotide. And that is basically this e to the qt is effectively the solution to the system of differential equations describing this Markov process model of evolution over infinite times, where basically we're multiplying this matrix by a particular time that is being elapsed. So this is the matrix form for these substitutions, and then this is now scaling it by the corresponding branch length. And then this is this geometrically fall-off distribution, which we're going to look at in our phylogenetics lecture with Chooks-Cantor and Kimura models, so that all of these entries have closed form solutions. So basically, we're going to be looking at this rate, and then this derivative is, in fact, the rate that we're looking at. And of course, there's a lot of hairy math for how do we actually estimate Q. We can collect as many alignments of known protein coding sequences, that's the training data, and then consider the probability of the training data as a function of Q, where basically the likelihood of Q is computed as a probability of the training data, and then these parameters Q and whatever elapsed time T, using the Felsenstein algorithm that we're going to talk about on next week. And then we're going to choose the Q that maximizes the total probability of having that likelihood Q of observing the training data. So we're basically choosing the parameters that maximize the likelihood of the training data. So there's many maximization strategies you can use. You can use expectation maximization. You can use gradient descent, simulated annealing, spectral decomposition, and others. And the branchlet can also be optimized in exactly the same way. Simultaneously, while optimizing the matrix, we're optimizing the branch length. And then we're also estimating the non-coding model similarly with random non-coding regions as training data. And given that the vast majority of non-coding regions are going to be not coding rather than some missed genes, that gives us enough power to estimate. So we can now use this generative model of codon evolution to compute the probability of a given alignment marginalizing over all possible ancestral sequences using Felsenstein pruning algorithm that we're going to see next week. So given a protein coding alignment, the probability of observing the leaves across all of these codons multiplied with each other, given the time elapsed, is 1 over 10 to the 117. And the probability of observing the same leaves here is 1 over 10 to the 275. So that means that if I simulate alignments randomly according to this model, I'll get this exact alignment only once every 10 to the 117 samples. Okay, so that's why that probability is too small. But the way that we're going to be computing whether something is coding or non-coding is by taking the ratio between these two models. We're going to basically ask, with what probability can I obtain this exact alignment under the coding model versus under the non-coding model? And same thing here, that exact alignment under the coding model versus the non-coding model. And we're going to take the likelihood ratio between those as our p-value with which we're going to be, the log odd score with which we're going to be deciding whether this alignment is coding or non-coding. Okay, so who's with me so far on sort of how exactly we're doing these kinds of evaluations? So we're learning these matrices, and then we're applying these matrices along the tree, and then asking how likely is it to generate that exact alignment using a coding matrix or a non-coding matrix for both protein coding and for non-coding alignments. Oops. So 25, 50, 25, 0, 0. So carrying out this exact likelihood ratio gives us 10 to the 35 for the log odds ratio here, for the odds ratio here. So that basically means that this region is 10 to the 35 times more likely to be protein coding than non-coding. And that region is 10 to the 21 times more likely to be non-coding than protein coding. So that is an enormous signal for such a short region. And then that's our measure of confidence that the alignment is indeed protein coding. Okay. So we can now use this to revise genes, look at sequence read-through, but also look at excess constraint regions. And I want to make a small parenthesis here, because we've actually used this approach to now revise the protein coding genome of SARS-CoV-2. So what I want to do is walk you through some of that work, because it's kind of cool. So I don't know if you guys recognize this sequence. So this sequence is, if you look at the tiny little font here, 29,903 nucleotides of trouble packed in a single positive strand RNA. If anybody wants to take a stab at guessing what this is in fact the entire genome of SARS-CoV-2. This is the virus that is responsible for COVID-19 and that has us all teaching remotely instead of being in class together. That genome is known to encode a large number of proteins from a single polypeptide that starts here with an ATG, encodes 15 different proteins, and then stops with a TAA. Then comes the spike protein that you guys have all heard about in the news, starting with an ATG, and then stopping here with a TAA. Then comes this ORF3A protein, ATG to TAA, and then the envelope protein, ATG to TAA, and then this membrane protein, ATG to TAA, and so on and so forth. Okay, so you would think that with a genome this small, everybody would know exactly what's encoded in the genome. And indeed, there's a lot of knowledge about many of these open reading frames. But in fact, for some of them, it says no known function for ORF7b, no known function for ORF8, no known function for, ORF10, and so on and so forth. So basically, many of these have no known functions. So the question is, are they functional or are they not functional? So what we basically did is use the exact same method of detecting protein coding versus non-coding signals. And then we applied it to a subset of the coronavirus genomes that are very closely related to the current COVID-19 genome of SARS-CoV-2 and the SARS 2003 virus, SARS-CoV-1 or SARS-CoV. And what you can see here is that the new COVID-19 genome is in fact a distant relative of SARS 2003. That lineage here is actually quite distantly related. And as soon as you get outside to MERS, these genomes just don't even align some of the ORFs at all. And then the other coronavirus genomes that are known to infect humans are in fact common cold viruses, 1, 2, 3, 4, which are largely benign and moving around this very frequently. So that's the phylogeny that we're going to be using. And then using this phylogeny, we can construct genome-wide alignments and then evaluate the protein coding constraint in each reading frame of the SARS-CoV-2 genome. And you can see here this frame shift site that switches the conservation from the second reading frame to the first reading frame in the middle of this part of the genome. And that's happening, That's happening here in the middle of the Pol2 protein. Basically, there's a slippage sequence here. GGGTTT, which basically causes the ribosome to slip. And instead of terminating at a stop codon here, TAA, it will then shift by one and then translate all of that, which is actually the polymerase protein. So that's a trick that the genome uses to sort of balance the rate with which the later proteins will be translated. So what we found using this is that many of the genes that everybody knows and loves in the first part of the genome here are in fact indeed amazingly well conserved. And many of the subsequent genes are in fact teaching us all kinds of cool messages. For example, we're seeing that in the S1 protein, this is the spike protein that basically attaches and then enters first attaches to the ACE2 protein and then enters the human cell by fusing the two membranes together. That is actually, very strongly, very fast evolving, and at the nucleotide level, but not at the protein coding level. You can also see this other protein here, which is protein 8, is in fact also super fast evolving at the nucleotide level, but very clearly conserved at the protein coding level. So you can use this to basically now revise the annotation of the SARS-CoV-2 genome. And what we're finding is that, for example, this ORF10 that has been annotated as protein coding is in fact in a perfectly conserved or a nearly perfectly conserved region, which shows very strong nucleotide level constraint. And the reason for that nucleotide level constraint is that it overlaps an RNA structure. But in fact, you can see that it has an internal stop codon and it has multiple disruptive substitutions here. You can see this ORF8, which is super fast evolving at the nucleotide level, but is in fact clearly evolving as a protein coding gene in those two places. And you can also see a dual coding region within the nucleocapsid protein. So here's ORF9b, which is in fact evolving as protein coding in both reading frames, and ORF9c, which is in fact not protein coding. And you can also discover a new protein coding gene within another protein. So using these signatures, we can see that ORF3a has another overlapping ORF within it, ORF3c, that actually encodes another protein. So these evolutionary signatures that basically we've used to understand the fly genome, the yeast genome, the human genome have also been very helpful in revising the gene content of the SARS-CoV-2 genome. So we can also now turn to revising genes and discovering new types of evolution within protein coding regions in human and elsewhere. So we can now basically take this model of protein coding evolution and then build a hidden Markov model that looks for +1, +2, or +3 conservation, or -1, -2, -3 conservation. And then just, put a Markov chain or a semi-Markov conditional random field, which is a discriminative version of a hidden Markov model, and basically scan the genome for regions that are new protein coding. So here you can see a very nice example of protein coding evolutionary signatures being extremely strong. So 100% or 0, 100% or 0 as that posterior probability of being protein coding. And you can also see this non-coding region here, which clearly has no protein coding signal, but is just as conserved, if not more conserved, than the protein coding exons. By contrast, this other region here that also doesn't match a known exon is in fact extremely clearly protein coding. So we can use this to now go and discover new exons. So we can again also discover new protein coding genes in the human genome, similarly new protein coding genes, the fly genome, in the yeast genome, and also discover new patterns of new types of protein coding genes. For example, what we found is that there are some stop codons that are just completely ignored by evolution. Evolution seems to be caring about these regions as a protein, caring about that region as a protein, but not caring about these regions as a protein. So for most proteins, you see this very clear transition from coding to non-coding. But for some proteins, the coding signal continues past the first stop codon. And we've used this to basically discover a new mechanism of post-transcription regulation that actually allows read-through for hundreds of proteins in the fly genome and a handful of proteins in the human genome. We can also discover these RNA structures that are associated with the stop codon read-through and this excess conservation surrounding these stop codons. We can also find overlapping selection for protein coding exons. So here there are, basically we can measure not only what the protein coding signal is like, but we can also measure synonymous constraint within the third codon position of protein coding genes. And across all of these Hox proteins, we were able to find that in fact they encode many additional motifs and RNA structures overlapping their coding regions. And that actually causes their synonymous rate to drop to 0. So we can separately estimate the synonymous rate and the non-synonymous rate across different genes. And we can basically find regions where the synonymous rate actually drops to 0, suggesting that there's like simply no toleration for synonymous substitutions, indicating overlapping protein code, overlapping elements encoded within the protein coding signal. We've also used this to now interpret non-coding GWAS signals with new protein coding genes. So here's one example where there was a, remember in the GWAS lecture, I mentioned that 93% of GWAS signals are in fact non-coding. Well, this is one of them, but for this and another 118 genetic loci, we basically found that we can now predict new protein coding genes that exactly overlap this genetic signal, enabling us to now show that for all of these different types of functions, these protein coding exons are in fact probably what's being disrupted. Here's one example of this gene that is expressed, that is this locus that is associated with myopia, and this gene which is expressed specifically within the retina and within the eye, suggesting that this is probably the function of that gene. So that was all about protein coding functions. Let's switch now briefly to signatures of RNA, of motifs, of transcription factor binding sites, and of microRNAs. So going back to these different classes of elements. So for protein coding, we saw these codon substitution frequencies, and for non-coding, we saw these reading frame shifts. So what about RNAs? So when we talked about RNAs, we basically talked about these context-free grammars that can be used for generating RNA sequences. We can basically build a context-free grammar that gives us production rules for generating A spaced by U with S in the middle, U spaced by A with S in the middle. So these production rules are basically recursive and allow you to generate pairs of nucleotides that are parsed along here. So if you look at the parse tree for this particular sequence, you basically have this empty sequence, this S which became empty, and then this S which became A, emitting A on the left, and this other S which emits G and C on the right and the left. And then this S here which emits a C, an S and an S, which is basically a fork, which basically captures this RNA structure. So this is a context-free grammar, but a stochastic context-free grammar basically has probabilities associated with these production rules, enabling you to infer the probability with which this grammar emitted that particular sequence. So you can use this to basically emit pairs of nucleotides, but you can also use this to emit alignments of nucleotides. And that's a stochastic context-free grammar that's on a phylogenetic tree. And that's basically PhyloCSF. So that allows you to now generate the parse of not just a sequence, but a phylogenetic alignment along that phylogeny, which basically at every point emits a whole vector of alignment, aligned sequences. And then this and that are G and C, and they're emitted together. A and U are emitted together. C and G are emitted together. A and U are emitted together. So we can now, in a single sequence, use the terminal symbols as base pairs, and then the emission probabilities are the base frequencies in loops versus paired regions. But much more powerfully, a PhyloSCFG has terminal symbol, has alignment columns as terminal symbols, or paired alignment columns. And the emission probabilities are calculated from the phylogenetic model and the tree using, again, Felsenstein's algorithm that we're going to be talking about in the phylogenetic lectures. So instead of having a 4x4 matrix for single columns, for paired columns, we have a 16x16 matrix that is trained on known RNA structures and then applied to look for evolutionary sequences that evolve in the same way. So using these, we can now go and search for new RNA structures and families. We can basically find families with paralogous conservation when these are actually conserved in multiple locations and so on and so forth. So that's for phylogenetic stochastic context-free grammars as a way to discover RNAs. So let's now turn to motifs. Again, this should be a review of the regulatory genomic signal, where we talked about these evolutionary signatures for regulatory motifs, where in any one location, the motif is preferentially conserved, but the signature for motifs comes from the fact that they're conserved genome-wide. So across hundreds of locations in the genome, you basically have preferential conservation of known motifs versus random controls of similar properties. And that's one signature, the intergenic region preferential conservation. Another signature is the bias to be preferentially conserved within intergenic regions rather than within coding regions, whereas random motifs are more conserved within protein coding regions because protein coding regions are generally more frequently conserved. And also, true motifs are conserved upstream versus downstream, whereas random controls are conserved in both. So that gives us three different signatures for genome-wide conservation of regulatory motifs. And again, applying these signatures, we can search for k-mers that are preferentially conserved, and then expand these k-mers into longer motifs through these extensions. So we can use the same thing for discovering now individual instances of those motifs. So of course, the challenges in individual motif instance identification is that the motifs might actually move and they might actually be lost. So the evolutionary signature for individual motif occurrences is instead that if we search for the pattern matching the motif independently in each species, regardless of the alignment, we can basically ask what is the total branch length covered by all of the species that contain a match to that motif. So how does the stochastic context-free grammar come up with new RNAs? It seems like we'd only categorize RNAs based on grammars most likely to generate. So for the stochastic context-free grammar, the way that it works is that you learn a general grammar for RNA classes. And then you have a probability associated with emitting pairs of columns compared to emitting individual elements. And then you can look at the likelihood ratio between this sequence being parsed according to a paired model versus a single model. And the grammar, however, is general. It doesn't always need to recognize exactly that structure. It's just that every production rule that generates pairs of columns is more likely in an RNA structure, but the production rules that only generate single columns are more likely in the unpaired regions. So that's how it distinguishes paired versus unpaired. Ayush, I hope that answers your question.
Yeah, sort of.
Great, thank you. Okay, so that's for genome-wide conservation of motifs. And then this is for individual instances of motif occurrences, where we can now ask what is the total branch length over which the sequences are conserved. And that gives us 2.23 substitutions per site. So that basically means that this entire tree, measured in units of substitution per site, captures these branches, but doesn't capture this branch, that branch, and that branch, and that branch, because either the motif is lost or that sequence is not present there. So now we want to ask, is this branch length sufficiently high, despite the missing species? And we can actually ask whether this branch length is above what you would expect by chance. Notice that this is allowing for mutations that are permitted by the motif degeneracy, and it also allows for movement of the motif where they don't all have to start exactly the same location because they're searched independently in each of the species. And these are the short branches that are missing. So as we mentioned in the regulatory genomics lecture, we can now look at the signal, some of which might actually be noise, versus purely noise. How? By using motif-specific shuffled control motifs to determine the expected number of instances at every branch length score that we would expect by chance alone due to non-motif conservation. And then we can translate that into a confidence score as a fraction of the instances above the noise. So the way that we do that is that we produce a lot of control motifs that have similar properties as the known motifs. And then we ask how frequently are the control motifs of similar type reaching a branch length which is similarly high. And you can see here that at this location here, 30% of the signal could be accounted for by noise, suggesting that in fact we have 70% confidence. And then over here, it's more like 90% is actually pure clean signal. So the last signature that I want to talk about is the microRNA evolutionary signatures, to basically look for, only 60 to 102 microRNAs compared to 760,000 microRNA-like hairpins. So it's an enormous challenge of signal to noise. So even with a false positive rate of 0.5%, we would have 3,800 spurious hairpins. So we need specificity of 99.99%, so basically more than 5,000-fold enrichment. The way that we do that is by looking at evolutionary properties of aligned microRNA sequences. So as you know, microRNAs are basically these short regulatory RNAs that are produced by these hairpins and then incorporated into the Argonaute complex to target and degrade target mRNAs that have a matching 7-mer. So these microRNAs are encoded through very particular kinds of structures which are incorporated into this Drosha and Dicer machinery to basically go and cut target mRNAs. And if you look at this alignment, you basically see this characteristic shape of high conservation, low conservation, high conservation. And we can use this property as one of the signatures of known microRNAs. The fact that there's this one side of the hairpin, another side of a hairpin, and then the stem here, which is, or the loop at the end, which is less conserved, and then the stem outside, which is less conserved. So this conservation profile is one of the evolutionary signatures that we can use. And you can see here that true microRNAs have much stronger correlation with the conservation profile compared to random controls. And then you can also look at the minimum free energy of the consensus fold, which is lower for true microRNAs. You can look at the structural conservation index. You can look at structural features of the hairpin stability, the number of asymmetric loops, the number of symmetric loops, and each of those basically gives you an enrichment that we can combine now to get a total enrichment for true microRNAs. So combining these multiple evolutionary and structural signatures, we now have more than 4,500-fold enrichment for true microRNAs. Most of it is coming from the conservation profile, but also from the hairpin energy, from the arm versus loop conservation, and so on and so forth. So we basically can put these together into a decision tree, which basically allows you to now learn whether something is a microRNA or not by combining multiple decision trees. And that's what random forests are based on. We basically build many decision trees, any of which can select cutoffs and the direction of cutoff. Every feature can be reused multiple times, and you can use serially by and or in parallel by or. And then you can build that into an ensemble classifier through bagging of model averaging that basically combines the predictions. And then you can take the median of the predictions, and that gives you basically robustness and also an ability to evaluate feature importance. So that basically allows you to now go and discover novel microRNAs that you can compare to the known microRNAs. So here are some examples of novel microRNAs that are discovered and how they align. So we also were able to use that to discover new properties of antisense microRNAs. So here's an example within the Drosophila Hox cluster. That's the developmental cluster that constructs the body plan. And you can see that if this region is transcribed in the forward direction, then this microRNA inhibits those genes. If it's transcribed in the reverse direction, then there's an antisense microRNA that is actually inhibiting those genes. So on the sense direction, you basically have inhibition of this one, and on the antisense direction, you have inhibition of the other ones. In each case, basically serving as a switch between sense or antisense, each time having different targets. And the prediction from that Alex Stark made was that if you were to ectopically express this sense versus antisense microRNA, you would then lead to a morphological change in the Drosophila. And indeed, you can see that this flight balancing organ called an altir was in fact transformed into a wing just by changing the balance between the sense and the antisense microRNA. Again, serving as a switch where the sense one would be repressing this gene and the antisense one would be repressing the other gene. So that concludes our lecture for today. We basically talked about comparative genomics as a way to recognize signatures into genomes. So looking at nucleotide conservation and evolutionary constraint, looking at evolutionary signatures that span multiple multiple nucleotide positions, and focusing on the patterns of change, focusing on protein coding genes to basically revise the protein coding and the non-coding genome of human, of fly, of yeast, and more recently of the SARS-CoV-2 virus, and also signatures for RNAs, motifs, transcription factors, binding sites, and microRNAs. Just as a small parenthesis, we can use these signatures to also search for constraint within the human genome. So basically, we can ask whether the mammalian constraint matches the human constraint, and we can use that to, in fact, infer that there's additional constraint within active regions of the human genome. So if you look, for example, within protein coding regions, yes, of course, they are the most well conserved. But if you look now within regions that show activity according to binding of regulatory motifs or novel short RNAs, these are as conserved as some protein coding regions, which is basically, suggesting that indeed there's additional function within the human genome, which you can recognize just by comparing, human population variation. So here's one example where you can look at the bound versus the unbound motif instances for TCCF. And indeed, you can see that this signal drops dramatically for the bound motif instances, suggesting that this heterozygosity, which is the amount of substitution that you have within the human population, is in fact indicative of constraint. And again, here we're amplifying our signal because we're using multiple instances of these motifs. And if you look across all of these different motifs, you see that they're all showing more diversity within unbound motifs and within bound motifs, suggesting that whenever they're bound, there's more constraint leading to more rejected substitutions even within the human population. And again, you can also look at what are the specific biological functions that are associated with this increased constraint. And then indeed, there are specific biological functions that show this increased constraint. So next lecture, we're going to be looking at evolution specifically and how we can use comparative genomics to learn new classes of evolutionary events. And then after that, we're going to be focusing on phylogenetics and phylogenomics. So last poll, who feels that they've learned some stuff today? Awesome. Very cool. So 50-50-0-0-0. All right. Thanks, everyone. And then see you. Actually, I won't see you tomorrow. Tomorrow, we don't have a mentoring session. So if you guys want to chat, send me an email. I'm happy to chat. But you now have a few weeks to make progress on your mid-course reports. Thanks, everyone. Bye-bye.